ribosome. He said, "Oh, you don't know what a ribosome is?" And I said, "No." And he said, "Well, have you ever taken biology?" I don't know. I'd never taken introductory biology.
Howdy, welcome back to the Idea to Impact podcast, where we are bringing you stories and the voices from faculty across A&M to share their stories of their experience with technology transfer and commercialization. Today on our podcast, we have Dr. Larry Griffin, who's an associate professor in the Department of Biology. He's been at A&M since 1986. He has a Bachelor of Science from University of Utah, which is close to where he grew up in Montana, and he has a PhD in biology from Stanford. He's also very interested in the origins of science, especially Robert Hooke, who discovered and named the cell and was the first to describe elasticity and was a renowned inventor and architect. You're going to hear today Dr. Griffing talk about how he became to be a scientist, how his childhood influenced where he is today, and you'll learn a little bit about his research and what he does here at Texas A&M. So let's get started. Welcome back to the Idea to Impact podcast, where we are talking to faculty and community entrepreneurs and inventors about their commercialization and research practice and process. Today we have Dr. Larry Griffin, who is a professor in the biology department here at A&M. And I believe he has been here since 1986. Is that correct, Larry? Yep, 1986. So he is going to be talking to us today about his experience, and he's got quite a bit of knowledge. He's not just a scientist and an inventor, but he's almost a little bit of a historian. So we're going to hear a little bit about that. So Larry, why don't you start by talking to us about your early life? When is your first memory of being an inventor or a scientist or an entrepreneur?
Well, I've always been kind of visually oriented. I think kind of in pictures. And so I grew up in Montana, and one of the more beautiful things around in Montana were Yogo sapphires. We lived not very far from the Yogo sapphire mine, and we would go out and have picnics there, and we'd get these sapphires, these uncut sapphires, which are gorgeous. And then we also did other kinds of rock collecting, picking up quartz crystals and calcite crystals and beautiful, beautiful different minerals of copper, all the different colors of azurite and malachite and all those kinds of things. Beautiful. And iron pyrite, beautiful fool's gold, and gold itself. We used to go around to the old gold mines and look for gold. And I thought that was really cool. And so when I was like 3 or 4, I wanted to be a mineralogist. It was a big word. And it was studying these pretty rocks. I really thought those were pretty. And so I continued to be interested in science, but I wanted to start doing experiments. And so we had a place in town, not too far from my house in Great Falls, Montana, that was called the Mclaughlin Research Institute. And it was, this is a small town up in Montana, and there's like nothing up there. But the Mclaughlin Research Institute had one of the best scientists in transplantation biology in the world there. And the name was Ernst Eichwald. And he had all of these inbred lines of mice that were inbred in so that they were isogenic, and they had different histocompatibility antigens on them. In other words, the ability of something to be transplanted into a mouse was determined by these histocompatibility antigens, which in part discovered. And so I did a high school science project looking at histocompatibility on red blood cells, and that got me interested in biology. I was also kind of interested in biology from just my high school. One of the cool things was biology had beautiful things, and so visually thinking was easy to do in biology. In fact, one of the first pictures that I really remember thinking from a subcellular point of view on things that I do now was a picture of the Golgi apparatus by Hilton Mollenhauer. Hilton Mollenhauer was actually here for many years at Texas A&M, and I was lucky to be able to meet with him and collaborate with him on some projects when I first got here. So anyway, that was a huge motivation. And then I went to Oberlin College, thinking that I would be interested um, and I wasn't sure whether I wanted to do geology or biology or whatever. And while I was at Oberlin, I discovered I was thinking about ways things start. How do things start? And then how do they carry through? And I was really amazed by the operon model of genetic regulation and bacteriology. And so I did some projects in bacteriology there. But while I was there, I found out that it's an undergraduate college that doesn't really have research active faculty. I mean, they were
Right.
kind of research active, but they weren't kind of doing the stuff I wanted to do. So I did some research, and I narrowed it down that I was going to transfer to one of two different places. One was either University of Washington in Seattle or the University of Utah
The U.
in Salt Lake City. The University of Washington in Seattle had a guy named Leland Hartwell, who at that time was just beginning to figure out the cell cycle in yeast. And I called him up and I talked to him, and he said, well, he didn't have a very big lab. He wasn't quite sure if he wanted another undergraduate in the lab, that kind of stuff. And it turns out Hartwell won the Nobel Prize about 5 years ago for
Wow.
cell cycle analysis in yeast. And so if I did go there and he didn't shut the door on me, it would have been a really nice experience. But the place I did go to was the University of Utah, where Ernst Eichwald had actually gone, and so I was familiar with it, that kind of stuff. But I knew this bacteriologist there named Gordon Lark, and we were interested in how um, DNA replicated at that time, and I was interested in how DNA replication could feed into the process of development. And so here I was, like a junior undergraduate, and we were doing these genetic experiments in bacteriology up in his lab, and we were having a lab meeting, and we were talking about different biology aspects, and he was talking about something like the ribosome or something, and I said, well, what's a ribosome? He said, oh, you don't know what a ribosome is? And I said, no. And he said, well, have you ever taken biology? I don't know. I'd never taken introductory biology. So I was doing all this research. I'd never taken introductory. I actually never graduated from high school.
Right.
And so Oberlin took me as a early admittance kind of guy.
Right.
And so he said, well, you got to take introductory biology. And since you haven't really expanded your horizon, you need to think about doing some research in plants. And I thought, that was a drag. So what I did is
sciences. You've named every science I've ever heard of. Biology, bacteriology, geology. You've been all over the place.
All over the place. All over the place. And so when I started working on plants, I was also interested in that question of DNA replication and development. And I got into a nice lab, and the work that we did was actually published in PNAS. And then I got into Stanford working on plants. And I've been working on plant cell biology ever since.
Okay.
Wow. It happened to one of my sons. He was, one of those kids that by the sophomore year of high school, he was getting in trouble because he was so bored. He was correcting his teacher's math on the board and everything. And I won't name the university, but a university took him early for that reason. But it's a shame that they don't have those more often, because there are a number of people like that, who kind of need to go to the next level to really expand and explore where they are. By the way, trivia question.
Yeah.
What do the three schools that you mentioned, University of Washington, Seattle, University of Utah, and Texas A&M University, all have in common?
They all have 12th men or something like that?
Yeah.
No.
They were all places Michael Young was the president.
Oh, that's right. Yeah, that's right. Actually, the guy that was Michael Young's precursor, Gardner,
Yeah.
who he knew pretty well, had actually done an interesting recruitment program at University of Utah, where they got high schools that were interested in doing science down from the Northwest to
Hmm.
go to Salt Lake City and talk to other kids. And then they had a big program where they could go around and listen to little lectures and that kind of stuff. And that was actually one of the things that also motivated me to go to the University of Utah, was this high school experience that I had, where I was pretty much on my own. I got a bus from Great Falls and went down
Wow.
to
Yeah.
Salt Lake, and when I was like 16, and it was pretty cool.
It's beautiful there too. I've been there a few times. University of Utah. It's gorgeous. Yeah.
It's great for skiing too. I mean,
Yeah.
if you do skiing, it's really fun. And that
That
you
was
can,
Salt Lake
yeah,
City.
Salt Lake.
Yeah. Isn't
Yeah.
it like basically a big crater,
Yeah.
kind of, and yeah, it's gorgeous. Yeah.
So Provo. We've been up to Provo. We went up there for lacrosse, a lacrosse weekend. We played both BYU and University of Utah. It's beautiful up there. I
It
mean,
is.
at BYU, you're playing on this lacrosse field, and then you just look up and there's just like mountains. It was April, so the weather was beautiful, and you know, I imagine that like being in Montana really heavily influenced like where you are now in terms of what you decided to study and your interest in plants and biology in general. I mean,
Yeah, I was also in Montana. We had I worked on this dude ranch, as I mentioned,
so cool.
and the dude ranch is now owned by the Nature Conservancy as a grizzly bear preserve.
Hmm.
And so when I was growing up, we had all these grizzly bears running around, and I would be on horses and we'd see grizzly bears and all that kind of stuff. And so one of the things that I do in imaging here is environmental imaging. I was so interested in image analysis at the cellular level, I thought maybe we could translate some of that to the environmental level. And so I worked here with a bunch of people who were in marine biology, doing analysis of the corals at the Stetson Marine Sanctuary, at the Flower Gardens National Sanctuary, I guess it is. And they had a yearly assessment of the growth on this flower banks area of benthic organisms. But what they had done in the past was they just went through and they used transects and counted them. And I said, well, what you could do is you could do color analysis of them. And you could
Oh,
actually identify the different creatures and the different areas that they occupied by doing color processing of those images. And that was the first time that people actually had done color processes of corals. And so that paper that I published back in the uh, late 80s, early 90s it has been cited a lot in the marine biology area, because it was the first time people used color analysis to see how corals were like bleaching and
yeah.
area and that kind of
We're
stuff.
going to talk more about your work, and especially about the technology that you're commercializing and stuff. But I do want to talk a little bit more about you, because I do think you're one of those fascinating individuals that could probably talk about 8 million different things, from how to hog tie a cow to to image processing. But I think about people, when you think of science, everybody goes to Albert Einstein or something like that, or maybe Richard Feynman, or some of the more famous folks within the field of, for some reason, physicists stand out amongst in the mainstream. But these are people, especially Einstein, with the stories, that were visual thinkers, like you, and Tesla was like this too. They tended to see things in their mind, and they started to play with them in their mind because they could see them, and then they took it down into the more analytical elements and of that. What's your process when you're thinking about things? So you talked about how to use color analysis in this other field, but what have you ever thought about and tried to think about your own mental process and how you approach problems with this very visual mind that you have?
Well, I look at something and then I just start asking questions about it. And that's hard thing to do sometimes for people, to try to shed preconceived notions about
Right.
what you're seeing, and start thinking about it just in terms of what is there, not anything anyone else has talked about, or trained you to think about it in those terms. And so that way, the nice thing about biology too is that a lot of those visual components are lead to questions that have never been asked before.
Hmm.
And so the entry level into actually getting research questions in biology was really low. I
Yeah.
felt like, if you're going to go into astronomy, you have to know all this crap before you finally get to
Right.
ask good questions. But in biology, almost everything that was out there, if you had an entry level question that was not known. And I think some people are afraid of that. I'm kind of surprised by how many people, don't want to put themselves out on the precipice of the unknown and look out beyond that, because it is
Right.
a little scary out there
Right.
to divorce yourself from previous notions and then look forward out onto the vast unknown. It's kind of scary.
What do you think is the most scariest part for most researchers? If anybody's listening to this and they, might want to get to the precipice, but they're a little afraid. Is it career elements? Is it just the fear of the unknown?
Um, Usually by the time they've decided to do science and stuff like that, they're not too afraid of the unknown.
Uh-huh.
But unfortunately, I feel a lot of people go in a linear way. They get trained this way and this way and this way
Right.
and this way. And I've seen so many people, and I actually saw this kind of sadly when I was a program officer at the National Science Foundation, where they've developed this idea and they found the answer,
Mm.
and now they don't know what to do.
Right. Right. Right.
And so you don't get the grants anymore, and they go, what can I do if I don't have any money? Well, you've already solved your problem. We're not going to give you money for a problem
Right.
that's already been solved.
Right.
So it was kind of tragic that way. And I think a lot of people, I think that's the problem, is that they need to have a project that actually can branch out and they can look at it from multiple different dimensions. And I think one nice thing about being in biology right now is there's so many technological innovations that if you're looking at it at one level, it's easy to take it to the next level.
Mhm. Yeah.
Am I right in remembering from something I read a while ago that biology as a field now is also becoming more interdisciplinary with other sciences? For example, we mentioned physics before, that they're using some ways of thinking in physics in the biology fields and vice versa, that they're thinking about biology in terms of physics and things like that.
Oh, absolutely. I think that's a big difference now between when I was going to graduate school and
Mhm.
now, is that everything that we do is based on probabilities and statistics.
Right.
And I remember being taught when even while I was a graduate student, if you got to do statistics on it, it probably isn't worth studying, because, it wasn't clear result. It wasn't an absolutely crystal clear result
Right.
that everyone could see.
Right.
And so now we're in the world of probabilistic biology, and we have all these chemists that are doing it, all these physicists that are doing it. Biology has actually expanded into all these different areas. And I think the difference between biologists and all of these different disciplines is becoming less and less clear.
Mhm.
Right.
So are you working with anybody outside of biology then on any projects like say chemistry or physics, or do you have other grants that you're on outside of you know your work?
Yeah, so I do collaborate with people. In the past, I don't currently have a collaborator that's a pure physicist or a pure engineer. But uh, I do collaborate with Alvin Ye over in biomedical engineering,
Right.
and he is an engineer, and he works on making these amazing 2-photon microscopes over there that were used for some of our research. And so he is a collaborator on a T3 proposal that we have,
Right.
and so we're currently funded on that. And he's great to interact with, and we've just got a paper out where he's a co-author, and Holly Gibbs, who's a staff member of the Microscopy and Imaging Center, who's just excellent, is helping us with some lattice or some light sheet microscopy over at the Microscopy and Imaging Center. And she's an engineer by training. And so she's actually helped me. She's read some of my book, which is an introduction to imaging called Imaging Life. And she's read some of those chapters. And I actually I wrote this for biologists, and biologists in the past, maybe not so much in the future, but in the past have had a big aversion to math. They don't really want to do the math.
That's why they pick biology, right?
Yeah, that's right. It's a science, but they
No
don't have to do
differential
the math.
equations.
Yeah. All these bad partial differential equations. And all
Right,
that kind of
right,
stuff.
right.
And so I wrote this imaging text that really didn't have any math in it, but it tried to explain things rigorously. And so we talk about Fourier analysis and all that
Mhm.
kind of stuff,
Yeah.
but it's in terms of not in terms of a lot of equations. The equations come at the end of the chapter, or maybe in the
Right.
figure legends or something like that. The text doesn't rely on the equations. So it's um, pretty nice to have someone with that kind of background, where she comes at it completely from a mathematical point of view, a physics and
Right.
engineering point of view, and then says, well, how do they think of it in relation to the way that I've reconceived it, so that I
Mm.
could explain it without math to a broad audience.
That's a real gift to be able to do that. Some of my work is using mathematical modeling in social, like tax compliance in governments and in societies. And when you get so into the mathematics of it and you're actually really excited about it, then you have to write the paper, and you're writing it for a social science journal. You have to learn how to tell the story without the mathematics, but you have to know enough about the mathematics so that you can talk about it to someone who knows the mathematics. And those types of partnerships, it reminds me of Tversky and Kahneman, Amos Tversky and Danny Kahneman, who made a lot of strides in psychology. But Tversky was the big math modeling guy. Kahneman was the big survey type of guy, and they were able to really bridge that gap there. So it sounds like the more these disciplines are coming together, the more they're learning from each other, but everybody else is also benefiting too. So.
Mm-hmm. Yeah, and I'm always really find it fascinating. You mentioned all these different imaging tools that we have across campus. And I think it's really neat how I always thought growing up, when somebody's using a microscope or something in their research, it's in their lab, but you guys actually kind of reach out across campus and are using other people's technology and sharing resources, right?
Oh yeah, and we sometimes come together and contribute money. I know that there's a new light sheet microscope that's being constructed, actually being built over in the Microscopy and Imaging Center, and so everyone kind of pitches in, and they want the features of it. A modern microscope these days doesn't look anything like a microscope that sits on a bench.
Oh,
And
really? What does it look
oh
like?
yeah, it just looks like this big optical thing,
Huh.
and it might not even have any eyepieces in it because everything is video these days.
Mhm.
And so
Oh.
yeah, like a lattice light sheet microscope. I have a picture of it in my book. It doesn't look anything like a microscope. It looks like a vertical optical bench that has lasers coming in from the
I'm so
sides,
picturing
and a camera coming out the other side. It's just like different, and it's interesting because places like the Janelia Research Campus and other researchers are publishing open source diagrams and plans for all of these things, so that people can actually build them on their own, and
Hmm.
that's a lot cheaper than buying them from a microscope company.
Oh.
So the microscope companies are now making things for people that don't want to build them. So it's like a cryo-electron microscope would be really hard to build, and they are making these. If you look at a modern cryo-electron microscope, it looks like this great big closet, with doors closed.
Right.
There's nothing that you can operate on it, in terms
Right.
of little knobs or anything. It's all just automatic, and it's amazing what it does, and it's super expensive, but it's all automatic and different from when I first got an electron microscope back at Stanford.
Mhm. Wow.
So, still talking a little bit about your own interests, and then we'll get into some of the technology. I know I got an email from the Royal Society, and they said they want their picture back. Do you want to tell us a little about the picture behind you? Uh,
Oh yeah, yeah. So there's that picture is um, possibly Robert Hooke. So
and
I can.
for those who don't know who he was, could you give us a bit on him?
Yeah, so one of the more iconic images in the history of science is this picture of the cells of cork, where the first published pictures of cells and identified as cells. So he was the first person to give the name cells to what we currently
Interesting.
use that name for. So um, and some people would kind of diminish his contribution by saying, well, Cork is dead and he just started dead cells and he didn't
Mm.
really know what he was dying about. But you actually read his book, Micrographia, which was this super high selling bestseller scientific book from 1666 and actually 1665, just before the London fire. And he goes through and he talks about all the juices in the plant cells, and he's interested in how the stuff inside the plant cells goes from one cell to the next.
Mm.
And there's a lot of interesting material in there that isn't in the picture. And a lot of people, tend to think of it just in the terms of those pictures. They don't actually read the text behind the picture,
Mm.
which is kind of sad. So anyway, he was a polymath like a lot of those guys, and he was a professor of geometry at Gresham College and one of the founding members of the Royal Society. He worked with Boyle in developing the Boyle's laws
Gas law. Yeah.
of air pressure. He made the vacuum pump and that kind of stuff that were necessary in order to do the experiments. And then he moved on to build this microscope, actually to use the microscope at the behest of the king at that time, Charles II.
Hmm.
So, yeah, actually, it turned out if you look at the history, um, the original idea for the book was thought to be awarded to Christopher Wren, who was the guy that was the architect that developed, the St. Paul's Cathedral and that kind of
Mhm.
stuff after the fire, after the London fire. And he said, well, he didn't really want to do that, and so Hooke volunteered, and so he put this book together at the behest of Charles II. Anyway, it was a very popular book, and he got a lot of credit for that. But he also had in there some other things besides living things. He had information about how light was transmitted, and he had information about gravity, and he had information about stars, and so all of those things were things that could be brought closer through using lenses, or were a phenomenon of lenses.
And he was a
So
contemporary of Isaac Newton, right? He was a contemporary of Newton's.
yeah, so Newton was younger than he was,
Okay.
but when he made lectures, he was a demonstrator at Gresham College at the Royal Society. I think it was a weekly demonstration that he would do of some experiment. So he had to build all these instruments and do all
Right.
these experiments and all that kind of stuff. And Newton would come down every now and then and look at those. And so Hooke was a little upset by the fact that uh, some of his ideas, what he thought were his idea, well, which were his ideas,
Right.
were adopted by Newton, and then he wasn't given credit for them.
Right.
And so this originated with the idea of how light was traveling through space, and Hooke thought light traveled as waves, and Newton thought they traveled as particles, and that was the way that the particles could slow down and make a spectrum from a
Mm.
prism. But uh, Hooke thought it was vibrational interference, interference between waves, and so they kind of came to a truce about that with a letter from Newton saying that he only got as far as he did by standing on the shoulders of giants, referring to Hooke, presumably, but that was kind of a sideways compliment because Hooke had spinal deformity.
Mhm.
And it is a kind of a good reference, and it's actually emblazoned above the threshold of the Royal Society that we stand on the shoulders of giants when you go in, but it was a little
Right. Right.
little sideways.
Wacky.
Anyway, so when Hook died, there was no uh, portrait of him that was available in his rooms, but it had been recorded that there was a portrait of him in the meeting room at Gresham College, and it was right next to Boyle's picture.
So
And
having a
so
portrait was pretty standard, though, if you were that type of guy back then, right?
absolutely.
Okay.
Everyone had a portrait. Yeah. And so there is a record of the portrait being there. Uh, Even 7 years after Hooke's death, Hooke died in 1703, and there's a good record of it in 1710. But it turns out that just in 1710, the Royal Society was moving their meeting area from Gresham College to another place called Crane Court, and this was when Isaac Newton was the new president of the Royal Society. And the second kind of tiff they got into was on the inverse square law of gravity.
Mm.
Okay, so other people had suggested the inverse square law of gravity, but Hooke really brought the idea to Newton, and he wasn't given credit for it. And also, um, they were both challenged by friends. Halley, the guy that Halley's Comet is named after, challenged both of them to uh, figure out how, under the influence of gravity, you could get elliptical orbits, which was Kepler's one
Right.
of Kepler's propositions. So Kepler didn't prove it, but he wanted these guys to prove it. So in Principia Mathematica, which came out in 17 let's see no it came out in 1692 or so, um, um, no let's see 1687 it came out the first edition of Principia Mathematica came out in 1687, and the portrait that I have here in behind me has a little drawing that he's pointing to, and that drawing is actually probably a drawing that only Hooke knew about. It was an unpublished manuscript of demonstrating how you could get elliptical orbits from a constant linear force that
Huh.
tended linearly with the distance. It didn't show the inverse square, but it showed a linear force, which also was in Principia Mathematica. And so he did show elliptical orbits under the influence of of a central force, but it didn't go to the level of detail that Newton was able later to show. And in fact, Newton did show that you get an elliptical orbit, but it wasn't that inverse square gravity caused elliptical orbits. He said that if you had an elliptical orbit, then you could get inverse square relations. So it was kind
Interesting.
of the converse of the
Yeah. Right.
proposition, and he didn't correct that until the 1720s, and even then he didn't show it mathematically. So Hooke actually did a more direct demonstration of the proof of elliptical orbits with that paper that is an unpublished paper at Cambridge University.
And that paper has the diagram that you think is in that picture.
Yeah, yeah, that paper has a diagram that's in the picture, probably in the picture, and then there's other iconography in that painting. Besides just the fact that the person in the painting looks an awful lot like the descriptions, the physical descriptions of Hooke, in that John Aubrey, who was a friend of his, and Richard Waller, who is his biographer, uh, uh, wrote down.
And did you say that Newton had a hand in the reason why there was no official portrait of Hooke?
Right. So the rumor was that Newton had the portrait destroyed, but my point is that if the portrait was there at Gresham College and they were moving to Crane Court, all he had to do, and there is no record by the Royal Society that they ever owned a portrait of Hooke.
Oh.
Yeah. So I don't know who that belonged to. It was in the meeting room, but it was easy for him to say, "Oh, that doesn't belong to us. Let's not move it to the Crane Court area."
Right.
So it wasn't really aggressive. Well, it might have been aggressive, but it was completely passive aggressive in terms
Was it
of.
like burn the picture?
Yeah, he didn't go out and burn the picture. Yeah. Well, it's nice that he didn't burn the picture because now we still have the picture.
Right.
Yeah.
That's
Wow.
fascinating. That's absolutely fascinating.